In this module, we'll continue our search for the hereditary molecule. In the series of experiments that I will show you, we are going to use a phage, a bacteriophage. It is a type of virus. Let me first of all show you the nature of this particle or this organism. Viruses are made up of two types of macromolecules, proteins and DNA. Proteins make the core in which DNA is present. These viruses attach with the bacteria and they enter the bacteria and they multiply and they produce progeny. They kill the bacteria and more viruses are released that can infect other bacteria. This is basically the life cycle of a bacteriophage virus. However, back then people did not know what part of the virus entered the bacteria. So here we are going to talk about a series of experiments. In this experiment, people grew viruses in two types of radioisotopes. What are radioisotopes? Radioisotopes are basically, as we know, we have talked about this, that nucleus of an atom has specific numbers of neutrons and protons. The heavier isotopes have more neutrons than the normal naturally existing isotope. So here they used two types of isotopes. One of them was sulfur, which is present in the protein. The other is phosphorus, which is present in the DNA. So they grew these viruses, two types, same virus, they grew in two different media. One contained heavy isotope of sulfur, the other contained heavy isotope of phosphorus. So I hope you see where things are going. They used these bacteria to infect, they used these viruses to infect bacteria. So when they infected these bacteria, they gave it a very short time duration. Soon after these bacteria were exposed to these viruses, these bacterial cultures were put in the blender. So basically what the blender did, when it rotated, it knocked off whatever was present outside the bacteria. There are two choices we have. Either the protein went inside the bacteria or the DNA went inside the bacteria. Whatever molecule went inside the bacteria is the molecule that is responsible for producing progeny virus. And what would produce the progeny virus? It would be the hereditary molecule. So after these scientists, they had these, after they put these cultures in the blender, they put the sample in the centrifuge. And now what centrifuge does is basically the heavier material settles at the bottom. So if this is a test tube and we put it in the centrifuge, the heavier material will be in the bottom and the lighter material will be on the top. Bacteria are larger particles in our system, so they will be at the bottom, and the supernatant or the clear fluid will contain whatever else is left, or probably the virus, part of the virus that did not go inside the bacteria. So they measured the radioactivity. Where is the radioactivity? We have two samples. One sample had viruses which had sulfur, radioactive sulfur. The other sample had radioactive phosphorus. So if the pellet of, or the whatever is centrifuge at the bottom, the pellet, we call it pellet, if the pellet of sulfur was radioactive, it would imply the sulfur went inside the bacteria. However, if the pellet is a pellet of the bacteria which were exposed to viruses with the heavier radioisotope of phosphorus, if that pellet is radioactive, it would imply that our DNA molecule, since phosphorus is present in DNA and we have only two candidates, protein or DNA, our DNA is the molecule that went inside the bacteria. So it turned out, of course, that it was the bacteria or the sample that contained viruses with a heavier isotope of phosphorus, that pellet was radioactive. They didn't stop there. The scientists did not stop there. They did additional experiment. They allowed the bacteria to grow. They had centrifuged the bacteria and they saw that the sulfur bacteria, they allowed both the samples to grow. The sulfur bacteria, when they were allowed to grow, bacteria with containing heavier isotope of sulfur in within the virus, when they allowed it to grow, their progeny did not have, they were not they did not have radiation. They were not radioactive. The phosphorus containing bacteria, which had the heavier phosphorus atom in them, when they made progenies, when these viruses were allowed to grow, these bacteria were cultured, and successively these viruses, the next generations also had phosphorus. So this experiment, Alfred Hershey and Martha Chase experiment in 1944, basically was experiment that conclusively that convinced the scientific community that DNA is the hereditary molecule. In the next module, we will see some features of DNA. Now we have established the DNA is the hereditary molecule. There was a race to study and understand how this molecule functions. We'll talk about that in the next module.